Anyway, the good news is we will be back in class tomorrow. All right. Since school was canceled today because of that tropical storm, what I've done here, I put this lecture together as a video. So what I'm going to record for you is the lecture I originally planned on giving in class tonight. So, in addition to the PowerPoint, let me show you what you need for this. You can download these files, what I'm about to show you from, just click on the video lecture bulletin, and there'll be links to download the Chapter 3 lecture outline, which is 2 pages, as you can see here. And the chapter, hold on. Internet is being slow right now. Here we have the Chapter 4 outline. As you can see right here. All right. And things are being slow for me right now, but just bear with me. Let's just bear with it. It's being slow. I don't know if my internet is, no. This table here, which I'm going to explain to you how the 9th edition, not the 11th edition. I'm not but I'm going to go over with you and guide you and film this out in the video, so should be no problem with that. All right. Yes, there is something you will need to be turning in. These outlines and this table I showed you, you're actually going to turn these in to me in class next Tuesday. They give you a week to do this. And I will grade you for completing. In other words, taking the notes and answering the study questions on the outlines and filling in the table. The grade I will be given is completing the activity as instructed. So it's either you do it, you get full points, you don't do it, you get a 0. There's no in between on this. If it's complete, you get full credit. And I will assign a point total a little bit later. I have probably looking at about 25, 30 points right now. I'll make the final decision later. All right, you should have your Chapter 3 outline in front of you. Okay, the water, we're talking about water. We're talking about why water is so important for life. Why without water, life as we understand it would not be possible. Think about how NASA has launched all these awesome space telescopes, the Hubble, the James Webb. The purpose for the James Webb telescope, James Webb was a NASA administrator during the Apollo. He ran NASA during the Apollo missions when we went to the moon. So what it does is it's searching out exoplanets. It's looking for planets around other stars. It's designed for this. And one of the things that this telescope has been programmed to look for are exoplanets that have a signature of liquid water. Think about it. What's the significance of liquid water? Very simple. Where there's liquid water, there's life. It's as simple as that. And the Earth, our planet, revolves around the sun. We're not too close to the sun or not too far away from the sun. We're in this range called the Goldilocks zone. So this Goldilocks zone is areas around the star that planets that are in these areas can possibly support life because the conditions are perfect for water to exist in all three states of matter: solid, liquid, and gas. And that's a clue in the search for life. Now Mars at one time, the planet Mars, you know, the dead red planet, well, it may not be dead. It may have life, microbial life in an area underneath the surface of the soil. There would be no Martians probably walking around because of the ultraviolet radiation, because Mars lacks an ozone layer. Thing I'm going to tell you about if it wasn't for the ozone layer, we'd all be walking around with all kinds of skin cancer. I'm not trying to be funny. I'm just stating a fact. All right. Now, water's unique properties has to do with its structure, the structure that holds the molecule together. So let's talk about the structure of water. Water, water everywhere, but not a drop to drink. Have you ever heard that phrase before? Think about it. Water, water everywhere, but not a drop to drink. If a person was saying this, where would he be? Anybody have an idea? If you had all this water around you, but you couldn't drink it, where would you be? Well, you'd be on a raft in the middle of the ocean. We can't drink salt water. It's fresh water where I'm talking about. Well, let's talk about polar covalent bonds. It is two polar covalent bonds. Each hydrogen joins to the oxygen molecule by a polar covalent bonds. Let's recall, what do we mean by polar covalent bonds? No, it doesn't have to do with the poles. No, polar means that the electrons being shared are shared unequally. One atom in the bond, the oxygen, is more electronegative. Electronegativity refers to, the infinity an atom has for electrons, not only its electrons, but other atoms' electrons, especially in regards to a chemical bond. And you already learned there are two types of chemical bonds, covalent and ionic. Now, covalent bonding may be nonpolar, in which the sharing of electrons is equal, or unequal in the case of polar covalent bonds. So because of this unequal sharing, one side of the molecule, the oxygen side, has a slight negative charge. It's a very slight charge. Where the two hydrogens are sticking out, there's a very slight positive charge. Now overall, the molecule is electrically neutral because the amount of negative is equal to the amount of positive, but the charge is distributed unequally. That's why there's a negative side and a positive side. And this polarity allows water molecules to form what's called a hydrogen bond with one another. You're probably wondering, what's a hydrogen bond? Well, we talked about that in Chapter 2, but I'm going to refresh your memory. Hydrogen bond is simply where a hydrogen atom, you see this hydrogen atom right here? It's already covalently bonded to this oxygen. But remember, the oxygen has a slight negative charge. And the hydrogen has a slight positive charge. Well, the negative side of water molecules will be attracted to a hydrogen atom of a different water molecule, such as you see here. We represent a hydrogen bond with dots. It's not a covalent bond, okay? It's not a covalent bond. It's a very weak interaction. But when you have a whole bunch of water molecules in a given area and you got all these hydrogen bonds, each water molecule is capable of forming 4 hydrogen bonds with 4 other water molecules. Well, it becomes significant. And the significance is that the 4 emergent properties of water emerge due to this polarity. The polarity of the water molecules causes these 4 emergent properties of water, which make life possible. So these 4 properties are cohesive, moderate temperature, in other words, resist temperature changes. Water expands upon freezing. That's why ice floats. And water is a universal solvent of biological systems. So, biological molecules dissolve readily, for the most part, will dissolve readily into water. So the chemistry of life inside cells takes place in water. That's why water is so important to us. All right. Water molecules stick together because of hydrogen bonds. We call this sticking of water molecules together cohesion. Well, cohesion enables small columns of water to be drawn up from the roots to the tops of trees. As water evaporates from the leaves, it creates a pull or tension on the water molecules, causing them to be drawn up. The water molecules stick together by hydrogen bonds. That's cohesion. And yes, the strength of cohesion is enough that water can be drawn up even 400 feet in a coast redwood by cohesion. And this is strong, this tension or pull is strong enough to resist the force of gravity. Now, at the same time, water molecules are being drawn up. They're also sticking to the sides of the cell walls that they're being drawn up through these special cells called xylem. Xylem's a vascular tissue. They're like little pipes. All that's left are the cell walls. So at the same time the water is being drawn up, the water molecules are also sticking to the cell walls. That's what helps the water to resist the force of gravity. So adhesion is when the water sticks to other things. Cohesion is when the water molecules stick to each other. All right, as water evaporates from the leaves, it's being drawn up from the roots all the way up to the leaves. As you can see here, where the water molecules stick to each other, that's by hydrogen bonds, that's cohesion. Where they stick to the sides of the cell walls on their way up, that's adhesion. Now, you don't need to worry about this right now, but this just shows you the tissue through which the cells that conduct the water. This is what this is showing you. We'll talk about xylem tissue in chapter 35. I'll say more about it then. Okay, well, I don't think we're going to be watching any videos today. I'm sorry. That's just not going to happen. But it's cohesion. Let's talk about cohesion. Cohesion is why, especially at the surface of the water, the interaction between the water molecules creates surface tension, and that's due to the cohesion at the surface. And that enables insects to walk on water and for you to skip stones across a body of water. Water is very effective at moderating temperature. Water, and essentially water can absorb a lot of heat before its temperature will raise slightly, or it can release a lot of heat before its temperature goes down. And that's a very unique property. That's why if you live next to a very large body of water like a Great Lake or an ocean, if you go inland a few miles, it'll be hot, but next body of water, it'll actually be about 30 degrees cooler. All right. We have some definitions here when we talk about temperature or heat. We're talking about kinetic energy. The motion of molecules, that's kinetic energy. What we call heat is actually thermal energy, which is the random motion. Temperature is simply a measurement. It measures the average kinetic energy. If the molecules are buzzing around faster, temperature goes higher. If they're moving around slower, the temperature will go down. And when you transfer thermal energy from one body of matter to another, heat always goes from a hotter area to a lower area. That's what we call heat. I sort of interchange the two terms because it's pretty much what it is. A calorie, this is a unit of energy, is simply an amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. That's also the amount of heat released when 1 gram of water cools by 1 degree Celsius. So it's the same amount of energy. If it raises the temperature, the heat's being absorbed. If the temperature lowers, then the heat's being released. Now, when we talk about calories on food packages, we're actually talking about kilocalories. Those are actually kilocalories, not simple calories. Now, the metric unit, the metric base unit for measuring energy is the joule. And the joule is simply 0.239 calories, or 1 calorie is 4.184 joules. Joule is the metric unit. Calorie is not a metric unit. It's an older unit that's still used by biologists. Specific heat. Water has a very high specific heat. That's why it has such good temperature moderating abilities. Specific heat simply is the amount of heat that must be absorbed or lost for 1 gram of that substance to change its temperature by 1 degree Celsius. Well, water has a very high specific heat. It has to release a lot of heat before its temperature will even go down a degree, or absorb a tremendous amount of heat energy before its temperature even goes up. Because of water's ability to resist its temperature change, that's why it has a high specific heat. Now, metals, on the other hand, have a very low specific heat. For example, if you take a skillet and put it on the stove and turn it up, pretty soon, if you don't get your hand off that handle, you're going to burn. And when you turn the heat off, that skillet will cool down fairly quick. Now, suppose you boil a pot of water, let's say about 3 gallons, about a gallon of water on the stove. Takes a long time. You're going to make some spaghetti noodles because you're inviting everybody over for pasta. You turn that heat on. It seems to take hours to get that water up to boiling. Now, when you turn that water off and just leave the pot alone, it's going to take a few hours before you can safely put your hand in that water without burning it. See? That water resists changing its temperature. It has a very high specific heat. And it all ties back to hydrogen bonds, the breaking and reforming of these hydrogen bonds. Because in a gallon of liquid water, you have all these little hydrogen bonds breaking and reforming. That's why the temperature doesn't change readily. That's why the temperature doesn't fluctuate much. And it's at and take case in point, the Earth is 70% water. That's why life is possible here. There are no major temperature extremes due to the temperature moderating effects of the large bodies of water. So when hydrogen bonds break, heat is absorbed into heat is released when they form. All right, I'm having some trouble here. Technical difficulties. But here you go. Yeah, let's say it's a nice summer day in San Bernardino. It's just about 50 miles from LA. It's 100 degrees in the shade. Yeah, Los Angeles Airport, 75 degrees. Now, this just shows you that the Pacific absorbs a lot of the heat. It doesn't change its temperature too much, so that's why it's cool by the ocean. And when you get away, you move away from those temperature moderating effects. See, the ground, the soil, has a very low specific heat. So it heats up readily. But when the temperature goes down at night, it cools off readily. But however, at the ocean, there's not that much fluctuation in temperature between day and night. And again, it's because of the water's temperature moderating effects. Let's talk about evaporative cooling. Well, when you sweat, and when the sweat evaporates, you cool down. You literally cool off. Well, that's what we mean by evaporative cooling. When the sweat evaporates, heat is being released into the environment. Ice floats. And the reason why ice floats is that in solid water, ice, the hydrogen bonds are stabilized. And when they're stabilized, it pushes the water molecules further apart. So that's why ice is less dense. In liquid water, hydrogen bonds are continuously breaking and reforming, and the water molecules are actually closer together in the liquid state than in the solid state. And actually, water reaches its greatest density at 4 Celsius. That's a unique property of water. Nothing else behaves like that. And yes, if ice did sink, pretty soon all the bodies of water be frozen solid. But when ice floats, it traps whatever heat there is underneath it in the water, so it's actually warmer underneath the sheet of ice. Warm enough it doesn't freeze down there where the fishes are huddled. So in ice, the hydrogen bonds are stable. And as a consequence of the stability, they are pushed further apart, resulting in ice being less dense. Now, in liquid water, the hydrogen bonds are continuously breaking and reforming, and the water molecules are actually closer together. They're moving around faster, too. Water is the universal solvent of biological systems. Now, let's define a solution as a liquid that is completely homogeneously mixed. All the substances in this dissolved are completely mixed thoroughly. The solvent is what does the dissolving. Water is the universal solvent of biological systems. The substance dissolved, be it salt or sugar, would be the solute. Aqueous solution is any solution in which water is the solvent, such as a saltwater saltwater solution. Dissolve 10% salt in water. The salt would be the solute. The water is the solvent. And because the solvent is water, that saltwater solution is also in aqueous solution. Aqueous is a Latin word referring to water. Aqua, agua. So when you dissolve salt in water, the ions are separated because they're charged particles. And you notice that when the water molecules, when they form a sphere of hydration, in this case, all the hydrogens are pointing towards the chloride, and all the oxygens are pointing towards the sodium ion. Water is such a good solvent because of its polarity. Polar molecules dissolve very well in polar solvents. Now, an ionic compound, when it dissolves in water, each ion is surrounded by a sphere of water molecules, and this is what you call a hydration shell, the sphere of water molecules around a charged particle or ion. Now, water can also dissolve non-ionic polar molecules such as acetone, proteins, things like that. Now, here's some more vocabulary. The terms are hydrophilic, any substance that will play nice with water. Polar substances are hydrophilic. Hydrophobic substances are things that won't play nice, won't dissolve in water, such as oil, gasoline, fats. Fats are hydrophobic. And the reason why they're hydrophobic is because they have nonpolar covalent bonds, which means they don't play with water. But hydrophobic molecules are an integral part of cell membranes, which makes cells possible, maintains the integrity of the cell. It is the interaction between water and the hydrophobic molecules embedded in the cell membrane. But that's a topic for later discussion. I mention it now, but we will come back to that point at another time. All right. Most in organisms, chemistry involves dissolving of solutes in water. Now, solute concentration in aqueous solutions. I'm going to skip this part because I'm not going to worry about it. You can read about it on your own. I'm not going to test you or quiz you on it. And you may have an assignment dealing with it in Master in Biology. Do the assignment. Don't do the work. But I'm not going to test you on it. Our last topic, though, will be acidic and basic conditions. Acidity, alkalinity. Alkalinity is basic. And that affects, as acids and bases affect living organisms, especially inside of our cells. Now, you keep in mind that a hydrogen atoms are able to undergo a process called dissociation. What does that mean in plain English? Well, I'm going to go over that. I'll show you what I mean by dissociation. There we go. My board is acting up on me. I got my tablet here, and it's being a butt. All right, let's see here now. I want to make this line, try to make it a little. Just bear with me a minute. See if this thing will work now. But anyway, really? All right, screw this. What happens in a water molecule? Water molecules can dissociate and break apart. What happens is a hydrogen ion can leave the OH behind. We call that hydrogen ion, and the molecule that's left behind has a negative charge, so we call it a hydroxide ion. And then very often that extra hydrogen ions floating around will combine with another water molecule to form a hydronium ion. Well, for the sake of simplicity, we're going to just forget about, we'll just think of hydronium ion as a hydrogen ion. And we won't worry about that hydronium. Now, water molecules dissociate and reassociate, so they break down into hydrogen and hydroxide ions, and then they reform water molecules. And it's a reversible reaction, at which the rate of dissociation is equal to the rate of reassociation. It's a reversible reaction. So two water molecules, one will give up the hydrogen ion to form a hydronium and a hydroxide, and then it'll go vice versa. Now, this dissociation in a sample of pure water, this dissociation is very slight. It's very small. So in pure water, the dissociation is on the magnitude of many times small. But dissociation of water molecules, though, inside of a cell can affect the cell's chemistry. And it can be disastrous, to the point of death. All right. Now, in pure water, I mentioned to you that water dissociates slightly. It does. So you'll have small but equal concentrations of hydrogen and a hydroxide. Most of the water is associated, but with a little bit of dissociation, but these two still will be equal. Now, you can add in other solutes to pure water, and it can change the concentrations of hydrogen and hydroxide ions. And we call these acids and bases. Acids will raise the hydrogen ion concentration. Bases will lower the hydrogen ion concentration and raise the hydroxide. We use a scale called the pH scale to measure hydrogen ion concentration and to determine whether a solution is an acid or a base or neutral. Acid is any substance that increases the hydrogen ion concentration. A base is a substance that reduces hydrogen ion concentration, and as a consequence, it raises hydroxide ion concentration. A strong acid, like hydrochloric acid, a strong base, such as sodium hydroxide, dissociates completely into ions. Now, other acids and bases don't dissociate completely. They're called weak acids and weak bases. And they can reversibly release and accept hydrogen ions or hydroxide ions, and thus they minimize changes in pH, but they can still shift the balance away. Now, pH is, all you need to know about all this is that pH is the negative log of the hydrogen ion concentration. In other words, it's a measure of hydrogen ion concentration. That's what it is. That's all it is. It's all you need to worry about. It's a measure of hydrogen ion concentration in a solution. It's It's a logarithmic expression, base 10 log. You don't need to worry about this math here. You'll learn about that math in chemistry. So, a neutral solution, one that has a pH of 7, will have equal concentrations of hydrogen and hydroxide. Now, what does this mean in the plain English? A solution that is an acid will have a pH value less than 7. And the lower it goes, the more acidic that solution is. A basic solution, one that's a base or alkaline, has a pH value greater than 7. And the more it goes up, the more basic or alkaline it is. And most biological fluids actually range around neutrality, pH of 7. Although blood is slightly alkaline under normal conditions, pH of 7.35 to 7.45, saliva is actually about 6.8. Urine, under normal conditions, has a pH of 6. Gastric juice in your stomach has a pH of 2. The food being processed in the small intestine has a pH of about 8. All right, this diagram illustrates the pH scale. A pH of 0 is very acidic. And yeah, it'll eat your skin away, probably. Now, each 1 unit change represents a 10-fold change in hydrogen ion concentration. So, solutions below a pH of 7 have a higher hydrogen ion concentration and a corresponding smaller hydroxide ion concentration. Each unit change represents a 10-fold increase, if you go this way, in hydrogen ion concentration, or if you go this way, it'll be a 10-fold decrease in hydrogen ion concentration. At the same time, when you're decreasing one, you're increasing the other. So, we have a solution with a pH of 6, okay? And a solution with a pH of 4. Well, in terms of acidity, the solution with a pH of 4 compared to the 6 will be 100 times more acidic, or have 100 times more hydrogen ions. At the same time, with the increase in hydrogen ions, you have a corresponding decrease in hydroxide. So at the same time, the solution with the pH of 6 compared to the pH of 4 have 100 times more hydroxide, but as well as 100 times less hydrogen. That's how the pH scale works. Although technically we're just talking about hydrogen ions. Household ammonia, about 11.5. Bleach, oven cleaner, lye, straight up lye is at pH of 14. So acidic solutions have more hydrogen than hydroxide. Neutral solutions, these two concentrations are equal. Basic solution has more hydroxides running around and fewer hydrogens. Now, cells have ways to maintain pH at a constant level so there's no fluctuations in the cellular chemistry. These are called buffers. And buffers tend to be weak acids in their conjugate base. And they can tie up excess hydrogen ions or release hydrogen ions as need be to keep the pH at a constant level, which is 7. Now, I'm going to take a very short break. And when I come back, we're going to talk about Chapter 4. Hello, and I am back. Very good. Now, For this chapter, the couple things you're going to need for this are the chapter 4 lecture outline, there you go. All right, this is the chapter 4 outline. And you're also gonna need, I'm having fun with playing with this. Bear with me, please. It's all getting slow. I don't think it likes me or something. I don't know. You will need this chart. All right, you're going to need this chart. Now, you don't need the chart right this minute. For now, you just need that lecture outline. And I'll let you know when you need the chart. Let's get back to the lecture presentation. There we go. Oh, I don't think we... All right, I'm having some technical difficulties. Ah, there we are. That's better. I think I figured out my technical difficulties. Okay. We're talking about carbon. Carbon is all living organisms are made of molecules that contain mostly carbon. Carbon has some unique properties. It has 4 unshared electrons in its valence shell. And so it has a valence of 4. Carbon wants to form 4 bonds, and it will form 4 bonds with up to 4 different atoms. And that's why it can form large, complex, and various molecules of increasing complexity. Now, the molecules of life, protein, nucleic acids, of which DNA is one, carbohydrates and fats, lipids, they're all made up of carbon compounds. This shows this represents, this Tinker toy actually represents a molecule with carbon at the center, hydrogen, oxygen, well, not oxygen, something else, it doesn't matter. Organic chemistry is simply the study of organic compounds. In other words, compounds that contain carbon. Biochemistry studies the carbon compounds of life. Now, there are other organic compounds, hydrocarbons, gasoline, which are organic, but they would be incompatible with life if they were ingested. So, biochemistry is one branch of organic chemistry. Now, Stanley Miller did an experiment which showed that organic molecules, even organic molecules found with life, can be synthesized abiotically or without life. So abiotic simply means non-life synthesis of organic compounds. He did a very clever experiment. And he set up this apparatus. All right, this represented the primitive ocean. In this primitive ocean, he added some compounds, mixtures of ammonia, hydrogen, methane, compounds that were thought to be part of the early atmosphere. So he boiled the water vapor, he placed these molecules, simple molecules and zapped with electricity to simulate lightning. And then he would cool the mixture. Now, he discovered amino acids and even formaldehyde. Formaldehyde is a simple organic compound. He found things that weren't there before, which suggests that organic molecules can arise without life. And in the 19th, we'll get back to that point later. And another thing that we see in living organisms is that the relative abundance of these 6 elements pretty much the same, with carbon forming a large structural backbone of the biomolecules. Now, it's carbon's electron configuration. That's why it's 4 unshared valence electrons. That's why it'll form 4 bonds with other 4 different atoms. And it's these electron configurations of the 4 main elements in life. And each element will form a specific number of bonds and also kinds of bonds. Carbon has a valence of 4. It has 4 unshared electrons. Now, the shape of the molecule depends upon if the carbon forms single bonds with other atoms or if it forms what's called a double bond. It changes the shape of the molecule, single versus double bonds. Remember, we talked about how the s and p orbitals can hybridize or overlap, causing atoms to, causing electrons can move about in the other orbitals temporarily. Now, when double bonds, it locks the molecule in place, makes it flatter, more rigid. See, in methane, the carbon joins to each hydrogen by a single covalent bond. So it forms what we call a tetrahedron. This ethane molecule here is a double tetrahedron. But when you add a double bond, such as ethylene, it flattens the molecule. Now, it is the number of unpaired electrons in the valence shell that the number of unpaired determines the valence. The valence is the number of covalent bonds it will form. That's what valence means, the number of covalent bonds it can form. And if you know the number of unpaired electrons, which you can get from the periodic table simply by noting the column that it's in, it actually tells you the number of unpaired valence electrons. But for now, these are the only valences you should commit to memory. Hydrogen has 1 valence electron, 1 unshared pair, so it has a valence of 1, means forms 1 bond. Oxygen has 6 electrons in its outermost shell, but only 2 are unpaired. So it'll form 2 bonds, here and here. Nitrogen has 5, but it has 3 unshared pairs. So it will form 3 bonds. Carbon has 4, so it'll form 4 bonds. So electron configuration gives it its compatibility with why it's compatible with many different elements. And carbon hangs out, and its other 3 most abundant partners are hydrogen, oxygen, nitrogen. So their interaction, because of their different valences, gives us the architecture for biological molecules. Now, here's a case of carbon dioxide. Oxygen forms 2 bonds. It has 2 unshared pairs. Carbon forms 4 bonds, so it forms a double bond with 1 oxygen and a double bond with the other oxygen. See, carbon has 4 bonds. Each oxygen has 2. So the Honk rule is obeyed. Urea. I mentioned urea because urea was actually synthesized abiotically in the 19th century. Before this one chemist did this, it was thought that only living systems could synthesize organic molecules. Well, 19th century, this one guy synthesized urea without abiotically using inorganic compounds. And he formed urea. And then we have Stanley Miller's experiments during the 20th century. But what I want you to notice are that oxygen forms 2 bonds with the carbon here. The carbon has formed 4 bonds, 1 with this nitrogen, 1 with this one, and 2 with this oxygen. Look at the nitrogen. Each nitrogen has 3 bonds. Each nitrogen has 3 bonds. Each hydrogen has 1. So the Honk rule is obeyed. Now, let's look at some of the diversity from some of the organic compounds. We can see how big some of these chains will get. So it's a chain of carbon atoms basically forms the skeleton of most organic molecules. You have a chain of carbon, which is the backbone. Well, these chains come in different sizes and shapes. Now, ethane and propane are examples of natural gas. They burn quite well. Ethane is a 2-carbon compound. Propane is a 3-carbon compound. These are hydrocarbons. It's carbon and hydrogen. These are very high energy compounds. They combust. When they combust, they burn. Now, butane is a 4-carbon compound. This is an isomer, a structural isomer. It has the same molecular formula, but because the molecule branches here, it has different properties, so it gets a new name. This 2 refers to the fact that this is attached to the number 2 carbon, the branch. Sometimes there will be a double bond in the chain. And yes, if there's a double bond, you change the ending of the name. Worry about that when you take chemistry. Don't worry about it in here. But that's why the name changes, because there's a double bond. What this 1 represents here is that the double bonds between the number 1 and the number 2 carbon. Now you're probably going to ask me, how do you know which one's the number 1? How do you know which one's number 4? You know what? I never could figure that one out. So don't feel bad. And here, this is number 2 butene because the bond's between the 2 and the 3rd carbon. Number 2, number 3. And yes, the position of the bond does change the properties. Sometimes a skeleton will form a ring around the rosy. Cyclohexane and benzene, the main difference is this is all single bonds and cyclohexane has more hydrogens. Benzene differs from cyclohexane because of the double bonds, it has fewer hydrogens. When you add a double bond between the carbons, you cut down on the number of hydrogens. Well, remember the Honk rule. All right, so this is a hydrocarbon. They vary in length. They can vary in branching, straight chain versus branched, or even double bonds, or a ring structure. And we're not going to, this is the bummer. This is the bummer. I can't do this way. All right, we've been looking at hydrocarbons. We were looking at hydrocarbons. Hydrocarbons make up fats. Gasoline is a mixture of hydrocarbons. Oils contain hydrocarbons. So the organic molecules, carbon and oxygen, fats have hydrocarbon components. Your cell membrane. Cell membranes have hydrocarbon components. That's the hydrophobic aspect of cell membranes. And hydrocarbons burn very well and release tremendous amounts of energy. All right, these are adipose tissue. These are fat droplets. Fat contains hydrocarbon chains. And we'll get more into that when we get into chapter 5 next week. Actually, I will cover chapter 5 on Thursday, so I hope you get this viewed before then. Isomers. Compounds that have the same molecular formula but different structural arrangements of their atoms. So they have different structures, which gives them different functions and gives them different properties. Now, a structural isomer, such as butane and 2-methylpropane, those were structural isomers. They have different covalent arrangement of their atoms. Let me go back there. I want to show you that. Or, yeah, these two are actually structural isomers. There's different covalent arrangement of the atoms. These are structural isomers here. These are structural isomers. I hope you get the idea. Cis-trans isomers, they have the same bonds but differ in the arrangement of the atoms. So the atoms they have the same bonds. So the bonds haven't changed, but the atoms differ in their arrangements. The carbons have the same arrangement, but the other atoms are rearranged differently, comparing cis to trans isomers. And then enantiomers are isomers that are mirror images. There's a right-handed version and a left-handed version. And yes, there can be a difference in their properties depending on if it's right-handed or left-handed. There is a difference in their properties. Structural isomers, such as pentane, a 5-carbon compound, and 2-methylbutane, they have the same molecular formula, C5H10, but the atoms are arranged differently. Cis-trans isomers. Here, the X's represent an element, could be any element. The X's are on the same side. But here in the trans form, they're on opposite sides. The X's aren't. So that's what we mean by different. They have the same bonds, but they differ in spatial arrangements. And enantiomers, right-handed, left-handed. And yes, the different forms can have different properties, especially with regards to drugs, pharmaceuticals. Now, the right-handed version of the drug and the left-handed version of a given drug may have different effects. In other words, one is biologically active, the other is essentially a dud. It doesn't help. It doesn't hurt, doesn't help. All right, ibuprofen comes in a left-handed form, which actually reduces inflammation and pain. Well, I take the right-handed form and the left-handed form. And the right-handed form takes away pain. Left-handed form doesn't do nothing. Albuterol. This is what they put in those inhalers, those bronchial inhalers, to minimize, asthma attacks. Well, the right-handed form is the form that relaxes the airways so they don't quit constricting. The left-handed form doesn't do nothing. And yes, when they produce the drugs, they have a way to separate the ineffective from the effective, so the drug is effective. All right. Concept 4.3, a few chemical groups. All right. We're going to talk about functional groups. Get that functional group table and get it ready. And get it ready because we're going to be filling it out in a minute. Now, functional groups are a small cluster of atoms attached to a carbon skeleton that participate directly in the reactions between different molecules and also confer upon that molecule its distinct properties. Now, there's 7 characteristic groups that we're going to worry about because they're associated with living organisms and biomolecules. All right. Estrogen, testosterone are a category called steroids. Steroids are derived from cholesterol. They have a carbon skeleton in the form of 4 fused rings. So they have the same skeleton. But what is different about them is the attachments. These are the functional groups. This one's called a methyl group. That's called a hydroxyl group. That's a hydroxyl. Now, this is estrogen, which makes girls girly girls. Testosterone is what makes boys manly men. The only difference between these two is the position of these functional groups here and here. And the absence of a methyl group here. And here we have a hydroxyl, but here it's just an oxygen. That's what makes girly girl, and that's what makes this manly man. Now, functional groups are the components these are the parts of the molecule that are actually involved in the chemical reactions. Each functional group has its own unique properties. Here are the 7 that are most important in the chemistry of life. These are the 7 you have to learn and know. And that's why you're going to be filling out that table in a few minutes. Now, on your handout, Part D, the chemical groups most important in the processes of life. For now, what I want you to do is to put the symbol for the group, the structural formula for the group. Just do that under hydroxyl is the OH. Just put that there. Then we're going to go back and fill in that table next. Carbonyl group, and I'm going to make this hold on now. Give me a minute. A carbonyl group is simply a carbon double bonded to an oxygen, and then that carbon will be bonded to 2 other things. So just put the symbol as you see it here. And you can go ahead and attach the 2 lines here. The carboxyl group is this structure here. It's an oxygen double bonded to a carbon, which is bonded to something else, which is bonded here to a hydroxyl group. So COOH. The amino group is simply, the structure is this line NH2. So the nitrogen is bonded to something on the skeleton and 2 hydrogens. It looks like that. And the sulfhydryl group, SH, with a line here. Now we're going to go ahead and work on that table. Now, the chemical group, I want you to, again, write the structure. Where it says structure, write the structure. I'm making you do it twice. Yeah, I know. I want you to learn it. So the structure of the hydroxyl group is simply the line with the OH. A name of a compound that possesses this group would be alcohol. All alcohols contain hydroxyl groups. An example, ethanol. And dissolves in water. One of its functional properties dissolves in water. We'll come back to the functional properties in a minute. We'll leave that alone for now. All right, but it does dissolve in water. The carbonyl group is this oxygen double bonded to a carbon, and the carbon will be bonded to two other things. So you can go ahead and draw that structure there. The compound name for the carbonyl would be ketone and aldehyde. Now, what's the difference between a ketone and an aldehyde? It's very simple, the position of the carbonyl. If the carbonyl is away from the end of the molecule like it is here, then it's a ketone. If the carbonyl is on the end, how can you tell it's an N? Because there will only be an H attached to the carbon at the end. There won't be anything. Now, if it had an OH there, it'd be a carboxyl group. If it's just an H on the end, it's a carbonyl. If the carbonyl is on the end, then it's a aldehyde. Now, these two are actually structural isomers. Acetone and propanol are structural isomers. The reason why acetone is a ketone because the carbonyl is in the middle. The reason why propanol is an aldehyde because look where the carbonyl is on the end. Carboxyl group is this, there's your structure. The compound name is organic acid or carboxylic. It's an acid because it will donate that hydrogen ion to solution. And that's a characteristic of acids. They donate hydrogen ions to solutions. Carboxyl groups are associated with weak acids. Organic acids are also weak acids. The amino group, NH2, the compound name is amine. The example here is glycine, which is an amino acid. Amino acids are interesting little beasties. They actually have an amino group on one end attached to the central carbon. That's called an alpha carbon because it's the center. And then it has a carboxyl group over here on the other end. So sometimes these organic biomolecules have more than one functional group associated with them. Now, what's nice about these amino groups, they act like a base because they accept hydrogen ions. They function as bases, forming this structure here. All right, properties. The property of the hydroxyl group, it's polar. These are polar groups. They form hydrogen bonds with water, so that's why they dissolve in water. So now we're on functional properties. That was the hydroxyl group. The carbonyl group, functional properties. Sugars with ketone groups are called ketoses. Those with aldehydes are called aldoses. That's your functional properties. That's what you write. Carboxyl acts as an acid. That's what you need to write down. Amino group, functional property, acts as a base. All right, the sulfhydryl group, now the structure as a line represents a covalent bond, so it's covalently bonded to a skeleton, SH. We call that a thiol. An example of that is the amino acid cysteine, which contains about 3 different amino acids have sulfhydryl groups. They participate in very strong covalent bonds. Phosphate group. Yeah, you draw, that's the structure. Compound name, organic phosphate. Organic phosphates are associated with DNA and RNA, and also certain alcohols. Methyl group, the structure is simply aligned, CH3. The compound name, just put methylated compound. Anything that contains a methyl group is methylated. Such as this structure here. This is this compound's called 5-methylcytosine. And anybody guess what the 5 stands for? I'll give you a hint. The methyl group's attached to the number 5 carbon. Now your question is, how do you know that's the number 5 carbon? I don't expect you to know that. That's my answer. All right, properties. Sulfhydryl group and two sulfhydryl groups can react, forming a cross-link that stabilizes protein structure. That's your functional property. The phosphate group, it contributes its functional property. It contributes negative charge. When it's attached, it enables that molecule to react with water to release energy. ATP, adenosine triphosphate, contains 3 of these phosphate groups. And ATP releases energy to chemical reactions in cells. Methyl group, functional properties, affects the expression of genes. A methyl group attached to a gene will silence it. When it's removed, the gene is active. And also attached to sex hormones. Determines whether you're going to be a girly girl or a manly man. Estrogen versus testosterone. ATP, adenosine triphosphate, that is the energy currency of the cell. It's a nucleotide, organic molecule with a smaller molecule called adenosine, which is attached to 3 phosphates. Now, ATP is used to release energy in cellular chemical reactions. And for now, that's what you need to know. And this is the ATP, and ADP can be recycled back into ATP by other reactions. So it can gain energy as well as release energy. And this, ladies and gentlemen, concludes the lecture. Remember, fill out those note sheets and submit them to me for a check. If you do the work, there's not a whole lot to do. Actually, you'll get full credit. And each outline will be worth 15 points. So you need to do it. All right. I will see you in school on Thursday and plan on I got to pull up the syllabus, but we will start with chapter 5.